So perhaps if we move on to, a big topic of discussion in radiopharm has been alphas versus betas. It's been a big topic. Obviously, the majority of your programs to date have focused more on the beta side, and particularly, with your therapy, copper-67 based. But recently you guys announced moving into a targeted alpha program with actinium-225. So it'd be really good, I guess, to understand the strategy behind that, but also the status of where you guys are at.
Yeah, it's a good question, Mel. It's we find we're a radiopharm company. Now, we've been doing this for a long time. I've been involved for 11 years. And we just want to produce the best therapies. And in radiopharm, it's a little more complex. It really is. It's not a small molecule that you just put in a patient. There's all these moving parts. But the market is suddenly talking isotopes.
Yeah.
And we find that fascinating. But the market lead at the moment are betas. That's where we're at.
That's the proven.
Right? But there's only 2 products on market. And we think about maybe the Algeta experience, Sofigo, is an alpha.
It's an alpha, yeah.
And we've got a lot of colleagues that were part of that Algeta group, including Thomas Rumdahl, who was the CEO, is on our board. So we're very close to that. Alphas are not to be messed around with.
Yeah.
We have found, and I've actually had feedback just last week, that big pharma's jumped in and making mistakes that we were making 10 years ago. And they've gone in and gone hard on particular things, not realizing that at the end of the day, you need to treat a human that needs to stay alive.
Yeah.
All right? And I've heard, preclinically, we get these amazing results with alphas. Yeah, but it's a mouse with a xenograph and makes the xenograph disappear. The mouse doesn't live any longer because then, you're doing this experiment. The human has to live and has to live with a better quality of life. This is what we're trying to share actually with the industry. This has to be the outcome. It's not about the isotope. It's about the whole product and treating it so you have great effects, right? It's highly efficacious. So you treat the cancer. And the human, right?
It's got to be super safe, right? That's the hard part.
and it's amazing. So if you look at the comparison of our data, right, in the secure trial, where you see the tumor shrink or disappear in the case of the complete response, and then you image much later and you see the tumor, it's gone, right? And you see all the offsite targeting, it's all still there.
Yeah.
So salivary glands, lacrimal glands, everything is still there. Right? You see when you treat with an alpha, everything is gone. And I don't know, those kind of glands kind of important.
Yeah.
Right? So particularly when you're looking at quality of life. And I find the industry has just jumped on board. So at the moment, as far as we're concerned, alphas are an experiment.
Yeah.
The supply chain and the clinical
every
practice,
part of them, right?
Every part of it.
Yeah.
I saw recently, and it was good that we're trying to look at this and expand the area. But you had, with dotatate actinium going into, say, breast cancer, early stage breast cancer. So we know that product binds to the pituitary gland.
Yeah.
Hitting the pituitary gland with an alpha is probably not going to be a good outcome for people.
Yeah.
So maybe starting to think it's not about just destroying the cancer. It's trying to find what we call the Goldilocks zone. Not too hot, not too cold, just right. So you're able to get that maximum effect on the lesion, but minimal effect on offsite targeting. And in this area of targeted therapies, we need to consider that.
Yeah.
Incredibly important.
And so is that where you kind of think of marrying your BIS PSMA, targeting specificity with an alpha with the hopes of kind of, I guess, building something a little bit more palatable, frankly.
Yeah. The hope, our position is, I don't think we're going to produce a better product than copper-67 SAR BIS PSMA, which has the balance of a beta with the targeting of the bispecific. I don't think we're going to do that. It's not going to get to earlier stage to treat a broader population. That is the sweet spot for us. That is our focus. But everyone was jumping on board. And when we looked at the 2 alphas, lead, which is just, everyone's now promoting 10-hour half-lives. It's just so complex, so hard. and the logistics supply chain around that and things. But we wish, for instance, the Aussie company, Advanced Cell, all the best with that. And it's a strategy. But there needs to be this combination. Alphas and betas need to work together. On the alpha side, we see that actinium has, at least today, the best supply chain, which is not a supply chain.
Exactly.
Which is not a supply chain. And there's experiments going on now to make rotatrons are capable of producing actinium. We hope it works out. And we thought, well, we've got the best, what we view as the best PSMA targeting agent ever invented. Right? From that perspective, the peptide-based ones. And so let's combine it with actinium. And let's see what we can do, because we seem to have this selective uptake into lesions, and that might be the reason why we have such a great safety profile with our copper-67. Let's see if we can have the same in actinium. Maybe we can dose less, have more selective uptake into lesions. Actinium is so destructive that we kill the cancers, but that offsite targeting is still going to be an issue. But currently, INT is the product being progressed. INT is not so good. So we thought, we can do this. We love science. We love chemistry. Let's make it. Let's do some preclinical work. And then let's see if it's worthwhile taking to clinic. But we think it's going to be in much later stage patients, much like the first alpha is, much later stage patients, and see whether we can have a beneficial outcome.
So you think of the two very much complementing one another in terms of, I guess, the continuum of care?
Yeah, so we view them as different products, even though they're the same products, just with the different isotopes, because alphas are different. We've got to treat them differently. They're not the same. They're not as friendly as betas. And as I said, everyone's on the bandwagon. Every venture capitalist now in the US knows, talks about alphas and betas. It's crazy what's going on. Make a molecule that works. It's the combination of the isotope and the targeting agent. It's just that no one's innovated the targeting agents.
Yeah, it's only one half thus far.
So they stick a actinium or whatever in it and then go, okay, let's just do the same. Let's not make the same mistake. We're going to have that strategy. We're going to keep them as different products. We're going to progress both in the clinic. We already are there with copper-67, SAR-bis PSMA. Incredible results because it was made that around that to achieve to be better than Pluvicto. That was our goal because that's the main player. Have an alpha strategy as well, and we'll look at alphas now. So we'll run all of our products because we can do this. We'll have an alpha strategy, a beta strategy, and obviously an imaging agent for all of those. And we can move into any targeting moiety we like. We can move into antibodies. We can stick with peptides and do a myriad of different things.